I want you to just picture a scene for a second, and I want you to really think about the sheer physical tension of it.
Yep. Set the stage for us.
Right. So it is the middle of World War II. We are in fascist Italy.
A terrifying place to be.
Exactly. And I want you to imagine a young woman locked inside her small, like totally unassuming bedroom. And outside her window, the regime is actively hunting down people of her heritage.
Systematically erasing them from society, yeah.
Right. But inside that room, she isn't just hiding. I mean, she isn't just waiting out the war in this state of paralysis.
She's working.
Yes. She is sitting at this makeshift desk, bent over a microscope, and she is painstakingly dissecting the microscopic nervous systems of chicken embryos.
Wow.
And she's doing this using a set of sewing needles that she literally sharpened herself.
See, the contrast there is just, it's what makes it so striking. Yeah, the macroscopic world outside completely tearing itself apart through, industrialized warfare and ideological hatred.
Yeah, absolute chaos.
But inside that localized, incredibly vulnerable space, she is deliberately focusing on the precise, microscopic building blocks of life.
It's just wild. Welcome to this deep dive. Today we are profiling the life of Rita Levi-Montalcini.
An absolute titan.
She really was. She was a pioneering neurobiologist. She later became an Italian senator for life, and she was the first Nobel laureate to live past the age of 100.
Which is an achievement in itself.
Truly. So our mission here isn't just to, list her achievements, but to trace the causal chain of a life that was fundamentally defined by defiance.
Right, because her whole life is just one massive act of defiance.
Exactly. We are looking at her biographical records, historical accounts of the era, and the primary scientific papers that, frankly, completely upended our understanding of the human body.
And the reason her story is so vital for you to understand is that she wasn't just a brilliant mind operating in some sterile, perfect academic bubble.
No, not at all.
She uncovered the fundamental wiring of the human nervous system while actively facing down this gauntlet of sexism, fascism, and anti-Semitism.
She proves that rigorous, empirical scientific curiosity can be, in and of itself, an act of profound rebellion.
Absolutely.
So let's start by looking at the environment that forged her, because the cultural context of early 20th century Italy is super crucial here. She was born on April 22, 1909, in Turin.
Right, and at that time, Turin was this massive industrial and cultural engine in northern Italy.
Like the center of everything.
Yeah, it was a city defined by production and intellect, but also by very, very rigid social hierarchies.
Totally. And her family background really perfectly encapsulates this specific intellectual tension of that era.
How so?
Well, she was born to Italian Jewish parents of Sephardic descent.
Okay, so a lineage with roots tracing all the way back to the Roman Empire.
Exactly. It carries this incredibly deep historical tradition of scholarship. Her mother, Adele, was a painter.
Ah, okay.
And her father, Adamo, was an electrical engineer and a highly capable mathematician.
So right away, you have this household that is fluent in two very different languages.
Yeah,
right?
what do you mean?
Well, you have the fluid, expressive language of art from the mother, and then the rigid structural language of mathematics and engineering from the father.
Oh, that's a great way to look at it. And you actually see that duality manifest clearly in the children.
Right, because Rita was a twin, wasn't she?
She was. She had a twin sister named Paola. And Paola completely absorbed the aesthetic side of the household.
She became an artist.
Yeah, highly respected artist. She was later known for these incredibly complex, geometrically precise aluminum sculptures.
Wow, so still blending the art and the engineering.
Exactly. It was an environment that essentially demanded intellectual engagement, regardless of whether you were looking at a canvas or a mathematical equation.
But the paradox here is that despite this rich intellectual environment, Rita's initial ambition wasn't to be a scientist at all.
Not even a little bit. In her teenage years, she desperately wanted to be a writer.
Really? A novelist?
Yeah. She idolized Selma Lagerlöf.
Oh, the Swedish author.
Right, the first woman to win the Nobel Prize in Literature.
Which, tells us a lot about her early mindset. Because a novelist's primary job is observation, right?
Right. You stand slightly apart from the world.
Exactly. You observe the human condition and you document the tragedies and triumphs of life. It's a passive, albeit deeply empathetic interaction with the world's pain.
Okay, let's unpack this, because changing career paths from a novelist to a neurobiologist because of a profound loss is like rewriting the entire genre of your own life story overnight.
It really is a massive pivot. What was the catalyst?
A massive, deeply personal trauma, A close family friend develops stomach cancer and dies.
Oh, wow.
And Rita watches this happen. She watches the absolute helplessness of the doctors, the inevitable decline of someone she cares deeply about, and it completely rewires her worldview.
She doesn't want to just observe anymore.
Exactly. She decides that she doesn't just want to document the human condition, she wants the agency to actually intervene.
So she decides to go to the University of Turin Medical School. That pivot is so profound because she isn't just changing her major, She is fundamentally altering her relationship with suffering.
Moving from the role of an observer to the role of a combatant.
Exactly. But she hits an immediate roadblock, and it isn't an academic one.
Nope, it's her father.
Right. Adamo Levi strongly discouraged his daughters from attending college.
He genuinely believed that an intensive higher education would somehow ruin their potential as wives and mothers.
Which sounds shocking to us now, but we have to analyze that resistance within the context of the era.
Yeah, it was a very different time.
It really was.
Mm.
Adano wasn't necessarily acting out of malice, right? He was operating within this incredibly deeply ingrained Victorian and early 20th century patriarchal mindset.
Like, that was just the water they were swimming in.
Exactly. Even in highly educated families like theirs, the prevailing sociological belief was that the domestic sphere was the only natural, biologically appropriate domain for a woman.
A professional career wasn't just seen as a distraction.
No, it was viewed as a dangerous, destabilizing force. The fear was that it would make a woman socially alienated and essentially unmarriageable.
So she isn't just arguing with her dad over the dinner table. She is arguing against the entire sociological consensus of 1920s Italy.
Which is a heavy burden for a teenager.
It's massive. And this is her first real crucible. She has to exercise this sheer, undeniable will to break through his preconceptions.
She doesn't take no for an answer.
Never. She argues, she pushes, and eventually she actually wears him down. He relents and supports her decision to enter medicine.
And that victory, I think, sets the template for her entire life.
How so?
Well, it taught her that authoritative structures, even those imposed by people you love and deeply respect, are not absolute.
They can be dismantled.
Exactly. They can be dismantled with enough persistent logic and will.
So she enters the University of Turin Medical School, and she completely thrives.
She's a natural.
Yeah, and it's there that she meets Giuseppe Levi.
Oh, a hugely important figure.
Huge. He was a neurohistologist who becomes her primary mentor.
Right, and Giuseppe Levi is a monumental figure in this story. As a neurohistologist, his entire focus was on the microscopic architecture of the nervous system.
Which, in the 1930s, was still basically a black box.
Totally. The nervous system was deeply mysterious. Scientists were just starting to use these complex silver staining techniques to make individual neurons visible under a microscope.
Like developing a photograph.
Exactly like that.
Uh-huh.
Levi taught her how to prepare these slides, how to look at cells not just as static little blobs, but as dynamic living structures.
And that sparked her obsession.
Yeah, her obsession with the developing nervous system, like how it grows from a single embryo into this staggeringly complex network.
So she graduates summa cum laude with her MD in 1936.
Talk of her class.
Top of her class. And she stays on at the university as Giuseppe Levi's assistant. So, everything is aligning perfectly for a long, prestigious academic career.
But then, the geopolitical reality of Europe violently interrupts.
Mussolini is aligning fascist Italy more and more closely with Nazi Germany.
And in 1938, the regime introduces the Manifesto of Race.
This is the moment the academic bubble just completely bursts.
Right. The Manifesto of Race
Yeah.
and the subsequent racial laws, they were designed to systematically erase Italian Jews from public life.
It wasn't just social ostracization.
No, it was a legal severing.
Mm-hmm.
Jewish citizens were barred from public schools, from government jobs, and crucially for Rita, from academic and professional careers.
Her university career is just instantly decapitated.
Overnight.
She is legally forbidden from stepping foot in the laboratory, from publishing her findings, from utilizing the equipment she had trained so meticulously to master.
The state is essentially telling her that her intellect is suddenly illegal.
Which is a terrifying thought. For the vast majority of scientists, that is the end of the line.
Well, of course. Science requires infrastructure, you know?
You need stuff.
Yeah, it requires funding, sterile environments, peer review, and highly specialized equipment. When the state strips all of that away, The logical response is to stop.
You pack up, you try to secure visas, or you go into hiding.
But she does the exact opposite.
Because she can no longer go to the laboratory, she decides to just build the laboratory inside her bedroom in Turin.
Which is an astonishing logistical and psychological feat.
Wait, I need to ask a logistical pushback question here on behalf of the listener because, wait, how do you even procure chicken embryos and maintain a sterile environment in a bedroom during a fascist regime?
It sounds impossible.
It sounds like MacGyver meets Madame Curie. Like, how did she actually do this?
It required intense, almost crazy resourcefulness. For the tools, she obviously couldn't buy surgical scalpels anymore.
Right.
So she took ordinary sewing needles and she ground them down on a whetstone to create microscopic points.
She literally made her own surgical tools.
Yes, and she
Yeah.
used fine watchmaker's tweezers.
Unbelievable.
She did manage to secure a standard optical microscope. But as for the subjects, the embryos, she would ride her bicycle out to local farms in the countryside and buy fertilized chicken eggs.
Didn't that raise suspicions?
Well, to avoid suspicion, she told the farmers she needed the extra nutrition for her family.
Oh, which wasn't a stretch given wartime rationing.
Exactly. Everyone was starving, so buying eggs seemed totally normal.
So she brings these eggs back to her bedroom.
Right.
She carefully cuts a tiny, tiny window into the shell. She operates on the microscopic chicken embryo inside, making these incredibly delicate surgical alterations, and then, this is wild, she seals the egg back up with paraffin wax to let it continue developing in a makeshift incubator.
An incubator she basically built herself.
Right. And the incredibly sobering detail here is that after she finished observing the embryos, her family would often actually cook and eat the remaining egg contents.
Because food was that scarce.
Yeah. It is the ultimate distillation of survival and science operating in the exact same physical space.
It really is. And the research she was conducting wasn't just busy work, just to keep her mind sharp.
No, she was swinging for the fences.
She was tackling one of the biggest unanswered questions in developmental biology at the time.
Which was?
How does a developing nerve fiber know where to go?
Right, because if you think about it, a human embryo starts incredibly small, but it has to eventually wire a brain to the tip of a toe.
Millions of connections.
It has to send out a nerve fiber, an axon, across vast cellular distances to connect with a very specific muscle. How does it navigate that journey?
Well, at the time, the prevailing theory was what we might call instructed growth.
Okay.
Scientists assumed the target tissue, say a muscle in the leg, was actively instructing the nerve, telling it exactly how and where to grow.
Like laying down a precise set of train tracks.
Exactly like that. But Levi-Montalcini, looking through her microscope in that bedroom, started to observe something completely counterintuitive.
What's fascinating here is how she actually tested this. She performed these delicate surgeries where she would amputate a tiny budding limb off the chicken embryo before the nerves had fully grown into it.
A tiny little microscopic amputation.
Right. And what she found was the crux of everything. She discovered that the nerve cells in the spinal cord still grew. They still multiplied.
Even though the destination was gone.
Yes. But because the limb was gone, because they lacked a target, they eventually died.
Exactly.
Mhm.
And this requires a fundamental shift in how we think about biology.
Wait, let me see if I am grasping the underlying logic here. If the nerves are still growing out even when the target is gone, that means the body isn't building a precise, pre-calculated map, right?
Correct.
It's basically overproducing nerves in a massive, chaotic shotgun approach, sending them out everywhere, and then just letting the useless ones that don't connect to anything die off.
That's exactly what it's doing.
That sounds incredibly wasteful and inefficient for biology. Why would an organism evolve to do it that way?
It's a brilliant question, and it's exactly the paradox she was uncovering. It seems inefficient, right?
Yeah, super inefficient.
But it is actually an evolutionary masterstroke of adaptability.
How so?
Well, think about it. If the nervous system had a rigid, hardwired map, any slight genetic mutation or physical change in the body's shape would cause the entire wiring system to fail.
Oh, because the tracks wouldn't line up anymore.
Exactly. By overproducing neurons and sending them out blindly, the nervous system ensures that every single piece of tissue that needs a nerve will absolutely get one.
It's a numbers game.
Yes. And the tissue then provides some sort of signal that says, you made it, you can survive.
And the ones that don't get the signal?
The neurons that don't get that signal trigger their own death. It's a ruthless pruning process that allows the nervous system to perfectly custom fit itself to the body as it grows.
So she realizes that the target tissue isn't a blueprint, it's a life raft.
I love that analogy. Yes.
It provides a survival signal. If the nerve doesn't reach the life raft, it undergoes programmed cell death.
Apoptosis, exactly.
And she is observing the fundamental mechanisms of life and death on a cellular level, while outside her window, a fascist regime is enacting those exact same principles on a societal level.
The irony is inescapable. The state is literally trying to cut her off to ensure she has no target or place in society so that she will just fade away.
Right.
And instead, she is deciphering the literal biological code of how connections sustain life.
It's poetry, dark poetry, but poetry.
She even documented this surreal period decades later in a 1997 documentary called Death by Design.
But, this quiet, localized rebellion in the bedroom couldn't last forever. The geopolitical situation deteriorated rapidly.
Things got much, much worse.
In September 1943, Germany formally invaded and occupied northern and central Italy.
Right, the Italian government had essentially surrendered to the Allies, but the Nazis took control of the north.
So it was no longer just about discriminatory academic laws, it was now about deportation and the Holocaust. Imminent mortal danger had arrived at her doorstep.
The Levi family had to abandon absolutely everything. They fled south from Turin to Florence, plunging into a completely different reality of survival.
They were forced into deep hiding, assuming false identities.
Which is terrifying. Their survival during this period was largely due to the immense courage of non-Jewish friends who risked execution to shelter them.
But even stripped of her identity, living in this constant terror of being discovered by the SS or fascist informants, she did not remain passive.
She couldn't. It wasn't in her DNA.
During the Nazi occupation of Florence, she actively sought out and made contact with the partisans of the Action Party. She literally embedded herself in the resistance network.
And we see her core philosophy manifest again, right?
Yeah.
The moment the environment shifted, she shifted her application of knowledge.
Yeah. When Florence was finally liberated by the Allied forces in August 1944, she didn't just emerge from hiding and wait for the war to end.
No, she went straight to work.
She immediately walked into the Allied Health Service camps and volunteered her medical expertise as a doctor.
The duality of that transition is staggering to think about.
It really is. A year prior, she was using watchmaker tweezers to look at a fraction of a millimeter of chicken tissue in absolute silence.
Right.
Now she is in a chaotic, bloody triage tent, treating devastating artillery wounds, typhus infections, and the massive trauma of frontline combat.
It demonstrates the immense bandwidth of her intellect and her resilience. She operated comfortably at the microscopic extreme of theoretical biology and the macroscopic extreme of wartime trauma medicine.
It reinforces her foundational belief that knowledge cannot just be an intellectual luxury.
Exactly. It is a tool that must be actively deployed to alleviate suffering.
So the war finally ends in 1945. The regime is destroyed. She returns to Turin, re-enters the university, and is finally able to resume her research out in the open.
A huge relief.
But what she didn't realize was that the work she had done in secret, in that bedroom, had already started to quietly cross the Atlantic Ocean.
And this is where the story shifts from a tale of survival to a tale of absolute scientific revolution.
Because even during the war, through these clandestine networks and neutral countries, she had managed to get a few of her bedroom research articles published.
Yeah, in academic journals in Belgium and the Vatican.
And in September 1946, one of those obscure articles lands on the desk of Professor Victor Hamburger at the Washington University School of Medicine in St. Louis, Missouri.
Now, Victor Hamburger was a giant in the field of embryology.
Like he wrote the book on it.
He essentially did. And he was fascinated by her paper because her findings, this idea that target tissue controls the survival of nerves rather than the initial growth of nerves, it directly contradicted some of his own published foundational work.
Now, in the highly ego-driven world of academia, a senior professor reading a paper by an unknown researcher that contradicts his life's work usually results in a dismissal or like an angry rebuttal.
Most of the time, yes. They just bury it.
But Hamburger does something extraordinary. He invites her to St. Louis for one semester research fellowship to basically prove it to him in his own lab.
Which speaks volumes to the integrity of Hamburger as a scientist. He cared more about the actual truth of the biological mechanism than his own ego.
So she accepts the one semester fellowship. And that single semester ultimately turns into a 30-year residency in the United States.
Because she shows up and delivers.
She really does. She arrives in St. Louis, sets up her equipment, replicates her bedroom experiments perfectly, and proves to Hamburger right there in front of his eyes that she is correct.
The nerve cells die without the target.
He immediately realizes she is a generational talent and offers her a permanent research associate position.
And this structural support, finally having a stable, well-funded laboratory, this sets the stage for the defining breakthrough of her career.
Right, because now that they know the target tissue provides a survival signal, the race is on to figure out what that signal actually is.
Exactly. Is it electrical? Is it chemical? Is it structural?
To answer this, we move to 1952. She travels to Rio de Janeiro, Brazil, to work with a scientist named Hertha Meyer at the Carlos Chagas Filho Biophysics Institute.
The trip to Rio was purely pragmatic. Hertha Meyer had perfected specific in vitro tissue culture techniques that Levi-Montalcini desperately needed.
In vitro meaning in glass, like a petri dish.
Right. She wanted to isolate the variables. She wanted to take the nerves out of the complex, messy environment of the whole embryo and study them in a controlled petri dish.
Because she was chasing a very bizarre anomaly.
A really weird one.
A former student of hamburgers had noticed something strange. If you grafted a piece of a specific type of cancerous tumor, a mouse sarcoma, into a chick embryo, the embryo's nervous system would just go completely haywire.
Nerves would grow wildly, rapidly invading the tumor tissue.
So in Rio, Levi-Montalcini sets up a controlled experiment. She places a small piece of the spinal cord from a chick embryo into a culture dish.
Okay.
And then, a tiny distance away, but not physically touching it, she places a piece of this mouse sarcoma.
The tumor.
Yes. And the visual observation she makes next is arguably one of the most famous and poetic moments in the history of cellular biology.
It truly is. It's iconic.
Tell us what she saw.
Well, she looks through the microscope after a period of incubation. She probably expected to see perhaps some mild nerve growth, maybe some random sprouting.
Sure.
Instead, she sees a dense, explosive halo of nerve fibers erupting out of the spinal cord.
Like a starburst.
Yes. And they aren't just growing randomly. They're growing frantically and directionally, reaching across the gap directly toward the tumor cells.
She described it in her notes with this incredible imagery. She said it looked like rivulets of water flowing steadily over a bed of stones.
It was an aggressive, undeniable biological magnetism. And because they weren't physically touching, it proved definitively that the tumor was secreting a soluble chemical substance that could travel across the medium and radically stimulate nerve growth.
Wow. And when she took this back to the whole embryos in St. Louis, she noticed a critical anatomical detail that locked the whole theory into place.
A smoking gun.
She observed that when the nerves grew toward the tumor inside the actual embryo, they heavily invaded the veins, but they almost entirely ignored the arteries.
This is a beautiful piece of biological deduction.
Let's trace the plumbing here because this is amazing. Arteries carry blood from the embryo's heart to the tumor.
Right.
Veins carry blood from the tumor back to the embryo. If the nerves are exclusively following the veins,
It structurally proves that the tumor is dumping a chemical into the venous bloodstream.
the nerves are literally swimming upstream against the chemical current to find the source.
Precisely. The tumor is hijacking the body's natural developmental pathways. It is sending out a massive artificial chemical signal that tricks the nervous system into building a dedicated vascular and neural superhighway to feed the cancer.
Here's where it gets really interesting. This realization leads to her seminal 1954 publication, wonderfully titled, and brace yourself for the academic jargon, In Vitro Experiments on the Effects of Mouse Sarcomas 180 and 37 on the Spinal and Sympathetic Ganglia of the Chick Embryo.
Catchy.
Very catchy. But in that paper, she officially gives a name to this mysterious chemical signal. She calls it nerve growth factor, or NGF.
Now, naming the factor was step one. But biological science requires physical proof of the molecule itself.
Right, you can't just say it's a chemical. You have to show them the chemical.
Exactly. She needed to isolate this substance. And for that, she partnered with a biochemist at Washington University named Stanley Cohen.
Rita Levi-Montalcini possessed the theoretical biological vision, but Cohen had the rigorous biochemical expertise required to break down the tissue and physically extract the protein.
They were the perfect team. Together, they managed to actually purify the NGF protein.
And this discovery caused an absolute earthquake in the biological sciences.
It fundamentally changed everything. Before NGF, the consensus was that embryonic development was mostly a matter of rigid genetic programming.
Like a computer executing a script.
Exactly. NGF proved that development is actually a fluid, dynamic conversation between cells mediated by these soluble chemical messengers.
But let's dig into the why of NGF for a moment, because this is where it impacts everyone listening. Why is a chemical discovered in a mouse tumor interacting with a chicken embryo so important to human medicine?
Because the mechanism is universal across vertebrates. NGF proved that the various systems in our body do not operate in isolated silos.
They aren't just doing their own thing.
No. It demonstrated that the nervous system, the immune system, and the endocrine system are constantly talking to each other, using these growth factors as a shared language.
So how does NGF actually keep a nerve cell alive, like mechanically?
Okay, think of it at a molecular level. A nerve cell has receptors on its outer membrane, specifically on the axon terminals, the far ends of the nerve.
The tips?
Right. When NGF binds to those receptors, it is internalized and transported all the way back up the long axon to the nucleus of the cell.
Like a messenger running back to headquarters.
Exactly. And once in the nucleus, it actively suppresses the genes that trigger apoptosis, which is the cell's programmed suicide sequence.
Wait. So the default state of a nerve cell is actually to kill itself.
Yes.
And it requires a constant active chemical signal from the outside just to stay alive.
Exactly. It's a profound paradigm shift. The default is death, and survival must be actively earned by making a successful connection.
That is mind-blowing.
Without NGF, brain cells and peripheral nerves simply wither and die. And understanding this completely revolutionized how we approach neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS.
Because it opened up entirely new fields of targeted molecular medicine, looking for ways to artificially supply these growth factors to halt the decay of the nervous system.
Exactly. And it was for this monumental field-defining discovery that Rita Levi-Montalcini and Stanley Cohen were jointly awarded the Nobel Prize in Physiology or Medicine in 1986.
It was a deeply deserved, if somewhat delayed, recognition. She was in her late 70s when she received the call from Stockholm.
Very delayed, but undeniable.
And with that prize, she became the fourth Nobel laureate to emerge from Italy's incredibly small but historically profound Jewish community.
Now, for almost anyone else, winning a Nobel Prize in your late 70s is the definitive end of the narrative.
Yeah, it's the capstone. You write your memoirs, you accept honorary degrees, you give the occasional keynote speech, and you enjoy a quiet retirement.
But the concept of retirement was fundamentally incompatible with her personality.
She was not going to play golf.
Definitely not. She had already been made a full professor at Washington University back in 1958. In 1962, she established a second laboratory in Rome, essentially working two full-time jobs on two different continents for years.
Flying back and forth constantly.
She directed the Research Center of Neurobiology in Italy. She formally retired from her academic posts in the late 1970s, but she just moved her focus into different laboratories.
She used the Nobel Prize not as a finish line, but as a megaphone and a launch pad for an entirely new era of research, working intensely deep into her 80s and 90s.
Which is just staggering stamina.
And her focus shifted to a completely different cellular structure, the mast cell.
This is where her ability to spot patterns across seemingly unrelated disciplines really shines.
Okay, tell me about mast cells.
Historically, mast cells were viewed through a very, very narrow lens. They are a type of white blood cell, and they are famously known as the culprits behind allergies and anaphylactic shock.
Oh, like when you get hives.
Right. If you get hives from a bee sting or your asthma flares up from pollen, that is your mast cells degranulating, bursting open, and releasing massive amounts of histamine and inflammatory chemicals.
Right, so traditional medicine viewed mast cells essentially as an evolutionary nuisance, right? Like a hypersensitive alarm system that mostly just caused uncomfortable allergies.
Exactly. But she starts looking at them in the 1990s and says, no, there is something much deeper happening here.
She hypothesized and eventually helped prove that mast cells are not just isolated allergy triggers. They are central coordinators of human pathology.
She showed they are deeply involved in chronic inflammation and, crucially, in how the immune system interacts with the nervous system to generate neuropathic pain.
And in 1993, at the age of 84, I might add, she identifies the crucial mechanism of how the body naturally tries to control these cells.
At 84.
She identifies an endogenous compound called palmitoylethanolamide, or PEA.
Endogenous meaning our bodies synthesize it naturally on demand.
Right. She discovered that PEA acts as a critical modulator. When tissue is damaged and mast cells start releasing inflammatory chemicals, the body produces PEA to act as a brake.
To calm things down.
Exactly. How does the brake actually work, though? How does PEA calm down a mast cell?
Well, without getting completely lost in the biochemical weeds, PEA essentially binds to specific receptors that downregulate the mast cell's hyperactivity.
It tells it to chill out.
Essentially, yes.
Yeah.
It stabilizes the cell membrane, preventing it from constantly degranulating and dumping more inflammatory fuel onto the fire. It's the body's natural mechanism for restoring balance after an injury.
And the reason this is so vital is that understanding PEA served as a massive catalyst for an entirely new era of medical research into the endocannabinoid system.
Right. The endocannabinoid system is a vast, complex network of receptors and neurotransmitters spread throughout the entire human body.
It regulates basically everything.
Yeah, it regulates homeostasis, it keeps everything in balance, managing pain, inflammation, mood, and immune response. And PEA is intimately connected to this network.
Because of her foundational work highlighting PEA in the 90s, today we have advanced liposomal PEA formulations being used clinically to treat severe chronic nerve pain and neuroinflammation.
And operating completely outside the traditional opioid pathways.
Which is huge. She basically opened up a new frontier of non-addictive pain management.
It perfectly illustrates her philosophy. She didn't stay in her designated neurobiologist lane.
Never.
She followed the data from nerve growth to the immunology of mast cells right into the pharmacology of the endocannabinoid system.
However, when you operate at the absolute cutting edge of translational medicine, when you move from theoretical biology in a petri dish to practical pharmacology designed for actual human patients, you inevitably enter a much more complex and dangerous arena.
And this brings us to a chapter that requires a very careful, impartial analysis. The Fidia controversy.
This raises an important question about the systemic friction between pure academic science and the profit-driven mechanics of the pharmaceutical industry.
It does. It's a very murky area.
So let's lay out the precise timeline and the facts exactly as they are recorded in the historical sources. Starting in 1975, Levi-Montalcini began cooperating with an Italian pharmaceutical firm called Fidia.
Her interest in working with Fidia was driven by her research into gangliosides.
What are gangliosides?
Gangliosides are complex lipid molecules that are heavily concentrated on the outer membranes of cells, particularly in the nervous system.
Because her entire career was built on the idea that external chemical factors can influence nerve survival and growth, she was highly interested in the theoretical potential of using these gangliosides to actually aid in nerve repair.
And Fidia had developed a drug based precisely on this concept. It was called Cronacial.
Right. It was a therapeutic mixture of gangliosides that were physically extracted from bovine or cow brain tissue.
The clinical intent for Cronacial was to treat peripheral neuropathies.
These are conditions where the nerves outside the brain and spinal cord are damaged, often causing severe weakness, numbness, and debilitating pain.
From a purely theoretical standpoint, supplying damaged nerves with the very molecular building blocks they need to repair their membranes makes logical sense.
It does. It makes perfect sense on paper.
And initially, the clinical data seemed to support the theory. Levi-Montalcini publicly supported the drug based on its biological mechanism, and early, supposedly independent studies suggested that Cronacial was indeed effective at treating these neuropathies.
But then a severe and terrifying clinical reality emerged.
Yeah. Patients who were prescribed Cronacial began reporting a rare but devastating side effect. They were developing Guillain-Barré syndrome.
And Guillain-Barré is a nightmare scenario.
It really is.
It is a rapid onset neurological condition where the patient's own immune system suddenly attacks their peripheral nerves, stripping away the myelin sheath.
It leads to progressive muscle weakness, paralysis, and in severe cases, it can be fatal if it affects the respiratory muscles.
The biological irony here is staggering. A drug specifically designed and marketed to heal peripheral nerves was triggering an immune response that aggressively destroyed them.
Right.
How does that actually happen? Like, how does a cow brain trigger an autoimmune disease?
It comes down to a concept called molecular mimicry.
Okay, what is that?
Well, the gangliosides extracted from the bovine brains look incredibly similar to the gangliosides on human peripheral nerves.
They're almost identical.
Exactly. In some patients, the immune system identified the injected bovine gangliosides as a foreign invader and created antibodies to destroy them.
Makes sense.
But because the human nerves looked almost identical to the drug, those same antibodies began attacking the patient's own nervous system.
The body got confused and attacked itself.
Yes, As the reports of Guillain-Barré began to surface, international regulatory agencies acted. In 1983, following standard pharmacological caution regarding severe adverse events, Germany entirely banned Crenachel.
And other European nations quickly followed suit, pulling the drug from their markets.
But the country where the drug was manufactured, Italy, did not ban it.
No, they didn't. The Italian regulatory bodies waited an astonishing 10 more years until 1993 to finally prohibit the sale of Crenachel.
And that decade-long delay is where the scandal really erupts.
Why did Italy keep a dangerous drug on the market for 10 years? Well, an extensive criminal investigation revealed a massive web of pharmaceutical corruption.
The investigation uncovered that Fidia had engaged in systematic bribery.
Wow.
They had actively paid officials within the Italian Ministry of Health to secure the quick initial approval of Crenachel. Furthermore, they paid to aggressively push the drug for the treatment of a wide array of diseases for which had absolutely no clinical testing or proven efficacy.
They basically monetized the theoretical science and used bribery to bypass safety protocols to maximize their market share.
Exactly. So the inevitable question becomes, where does Rita Levi-Montalcini fit into this web?
During the investigation, her long-standing relationship with the company was dragged into the spotlight, and she faced intense, highly critical public scrutiny.
Sections of the press even questioned whether her Nobel Prize was somehow tangentially tainted or influenced by pharmaceutical money.
Her response to the media was direct and uncompromising. When confronted, she stated, "Nobel comprato, non ne so nulla," which translates to, "Bought Nobel, I know nothing of it."
We must be absolutely clear and strictly impartial as we evaluate this based on the sources. The facts establish three things.
What are they?
First, she actively supported the underlying biological mechanism of the drug. Second, the drug ultimately caused severe harm to a subset of patients. Third, the company manufacturing the drug was engaged in deep systemic corruption and bribery.
This scenario highlights an incredibly complex dynamic in modern scientific research.
It really does. It explores how brilliant, rigorously tested lab work can become dangerously entangled when it transitions into the corporate sphere.
A researcher can identify a legitimate molecular mechanism, but when a corporation takes that molecule, mass produces it, illegally bribes regulators, and expands its use beyond the data to maximize profits, the original scientist is caught in the resulting fallout.
Exactly. We are not taking a stance on her personal culpability or what she may or may not have known about Fidia's accounting practices.
No, we are simply observing how the friction between academic endorsement and corporate malfeasance played out in the public eye.
And it is worth noting that this wasn't the only time her leadership roles faced complex scrutiny. Later in her life, in 2010, the European Brain Research Institute, an institute she had personally founded in 2002 to foster neuroscience in Italy, faced significant internal friction.
Parts of the scientific community levied criticism against her regarding what they described as her autocratic leadership style over the institute.
But weathering these intense periods of public and academic scrutiny actually fortified her for the final and perhaps most surprising act of her life.
Because she wasn't about to retreat from the public eye.
Not a chance.
She was about to step directly into the ultimate arena of public scrutiny, national politics.
On August 1st, 2001, the president of the Italian Republic, Carlo Azeglio Ciampi, appointed her as a senator for life.
To understand the weight of this, a senator for life is not just an honorary plaque you hang on the wall.
No, it's a real job.
It is an incredibly prestigious constitutional role in Italy, reserved for citizens who have brought immense honor to the nation through outstanding achievements in the social, scientific, artistic, or literary fields.
And crucially, a senator for life wields full, legitimate voting rights in the upper house of the Italian parliament.
They have the exact same legislative power as any elected senator.
And Levi-Montalcini took this civic responsibility with absolute, uncompromising seriousness.
Which brings us to the political absolute pressure cooker of 2006.
Oh, this is intense.
At the age of 97, she attends the opening assembly of the newly elected Senate. Italy at this time is highly fractured politically, essentially split right down the middle.
She openly declared her political preference, choosing to support the center-left candidate for the presidency of the Senate, Franco Marini, and throwing her support behind the government led by Romano Prodi.
The political math here is what creates the tension.
Right.
Prodi's government held an exiguous, razor-thin majority in the Senate.
Like barely holding on.
Exactly. Every single piece of legislation, every vote of confidence came down to a margin of 1 or 2 votes.
And sitting there, consistently casting the deciding vote that kept the center-left government from collapsing, was a 97-year-old neurobiologist.
Because her vote was so mathematically critical, she immediately became a massive, highly visible target for the political opposition.
Right-wing senators were infuriated, accusing her of unfairly propping up a government that lacked a true popular mandate.
The political attacks bypassed her policies entirely and went straight for her demographics.
They got very personal.
The far-right politician, Francesco Storace, publicly mocked her advanced age, actually sending her crutches to the Senate, heavily implying she was too old and frail to be making decisions that affected the entire country.
And the vitriol escalated even further. In 2003, she had to file a libel suit for defamation against the prominent comedian and political provocateur Beppe Grillo, who during one of his public shows referred to the 94-year-old Nobel laureate as an old whore.
The sheer ugliness of the attacks is shocking, but what is truly remarkable is how she responded to them.
Which was essentially to ignore the noise and do her job.
She responded with the exact same unyielding defiance she had shown her entire life. She treated the political bullies in the Senate in the 2000s the exact same way she treated the fascist decrees in the 1930s, by completely and utterly refusing to be intimidated, silenced, or erased.
She just kept showing up, taking her seat, and casting her vote.
To understand her fearlessness in the Senate, you have to look at the totality of her identity.
She had spent a century operating entirely outside the traditional power structures of society.
Think about it. She came from a Jewish heritage, but lived her life as a vocal, unapologetic atheist.
She was an independent woman operating in the fiercely male-dominated ecosystem of mid-century science, and then the equally male-dominated ecosystem of Italian politics.
She never married, and she never had children. She simply did not fit into any of the boxes that society usually uses to control people.
And she was incredibly clear-eyed about those personal choices.
When a journalist interviewed her in 2006 and asked about her personal life and the absence of a traditional family, she offered absolutely no apologies.
What did she say?
She stated, "I never had any hesitation or regrets in this sense. My life has been enriched by excellent human relations, work, and interests. I have never felt lonely."
It is a profound rejection of the societal narrative that a woman's life is somehow incomplete or hollow without a traditional domestic structure. She authored her own definition of a fulfilled life.
And what a remarkably long life it was. On April 22, 2009, she achieved a milestone that almost feels biologically poetic for someone who spent her life studying how cells survive.
She became the first Nobel laureate in history to reach the age of 100.
The city of Rome threw a massive celebration for her at City Hall.
She continued to work, think, and engage with the world for 3 more years after that.
She passed away in her home in Rome on December 30, 2012, at the staggering age of 103.
The sheer diversity of the tributes that poured in upon her death perfectly captures the vast scope of her impact across completely different sectors of society.
The mayor of Rome mourned her passing as a great loss for all of humanity. The Italian prime minister at the time, Mario Monti, paid tribute to her deeply charismatic and tenacious character.
Even the Vatican released a statement praising her.
Which is wild, given she was an atheist.
Right. Despite the fact that she's a highly public atheist, the Vatican spokesman lauded her immense civil and moral efforts, calling her an inspiring example for Italy and for the world.
But the legacy of Rita Levi-Montalcini extends far beyond official statements and dense neurobiology textbooks.
It really does. Her impact bled into the wider culture in some delightfully unexpected ways.
The cultural footprint she left behind is fascinatingly eclectic. For example, if you are a fan of the massive open-world sci-fi video game Elite Dangerous,
Great game.
as you fly through the galaxy, you can actually dock your spaceship at numerous orbital stations that the developers specifically named after her.
That is so cool.
In the botanical world, in 2016, researchers discovered a spontaneous new hybrid orchid and officially named it Ophrys S. Montalcini to honor her memory.
From space stations to orchids.
She even crossed into esoteric cultural spaces, participating in major public events organized by the Freemasons, specifically the Grand Orient of Italy, to discuss the intersections of science, philosophy, and peace.
So what does this all mean? When you synthesize all of this, the space stations and video games, the hybrid orchids, the political battles, the Nobel Prize, the foundational textbooks, it points to a legacy that absolutely refuses to be categorized.
She proved that a family and a legacy do not solely rely on genetics.
Her legacy is deeply encoded in the scientific method itself. Every single time a modern researcher looks through a microscope to understand how a neuron navigates or how an immune cell triggers inflammation, they are literally walking through the intellectual doorways that she forced open.
It is an almost unbelievably awe-inspiring arc. We began this deep dive looking at a young woman in 1938, hiding in a bedroom, cracking open fertilized chicken eggs with a sharpened sewing needle while a fascist regime marched in the streets below.
And we end it in the hallowed halls of the Nobel Academy and the Italian Senate.
Celebrating a centenarian who fundamentally rewrote the instruction manual for the human nervous system.
As we conclude our profile today, I want to leave you with a final thought, drawing directly from the title of her own autobiography, which she brilliantly named In Phrase of Imperfection.
I love that title.
We live in a modern culture that constantly demands perfect conditions before we take action. We convince ourselves that we need the optimal funding, the flawless business plan, the perfect workspace, or the exact right political climate before we can start our life's work.
We perpetually delay our ambitions, waiting for an idealized perfection that will never arrive.
But the reality of history tells a different story. Rita Levi-Montalcini did her most vital, world-altering, foundational work in the most imperfect, terrifying, and resource-starved conditions imaginable.
She used sewing needles when she didn't have scalpels. She used a bedroom when she didn't have a lab.
So the question for you to ponder as you go about your day is this. What imperfect conditions in your own life right now? The severe limitations, the frustrating roadblocks, the constraints you wish you could magically wish away. What if those exact imperfect conditions are actually the crucible you need to discover your own true potential?
Thank you for joining us on this deep dive into the extraordinary, defiant life of Rita Levi-Montalcini. Keep questioning the structures around you, keep looking for the connections, and as always, stay insanely curious.